Let's hit it. And welcome to Alzheimer Speaks Radio. I'm your host, Lori LeBay, and I'm so excited that you're joining us today. We are going to have a fascinating conversation as usual as we learn from people all around the world at all ages and stages of life. Stay tuned as we shift our dementia care from crisis to comfort. All right, here we go. What you think about, you will bring about. Well, hi, everyone, and welcome to Alzheimer Speaks Radio. If you liked our intro music, it's called Clarion Call by the Mark Arneson Band featuring Maya Dora, and you can pick it up on any of your favorite music platforms. For those of you that are new to Alzheimer Speaks, we're about sound information, not just sound bites. Our goal here is to raise all voices, big and small, from those diagnosed to those that care and serve them, to advocates and researchers and more. And today we're having a live show, so you can join the conversation if you'd like by calling in to 323-870-4602. That's 323-870-4602. And of course, I'd be remiss if I didn't thank our listeners. You guys are amazing by spreading the word around the world of our work here. So I hope you continue to like, click, and share what we're doing, because it's all about building a sense of community, collaboration, and comfort so that we can win this battle against Alzheimer's and all the other forms of dementia. Today, our talk is going to be a conversation with two researchers, and it's going to be all about hope in the arena of Alzheimer's disease. So I'm really, really looking forward to that. But before I go there, I always like to mention that our shows are all archived. We've been doing this since 2011. And so our next show on Thursday is going to be with Valerie, who is actually diagnosed with dementia. I think you're going to find that really informative. And a couple of our past recent shows, we had Maria's Place on, which is all about activities and engagement that is free. We talked about smart living and estate planning with Catherine Hodder. We talked with MD V.I.P. on their brain health quiz, and so many more. Like I said, everything is archived in there to grasp when you need it. I also want to let people know with memory cafes, a lot of them have not converted to virtual, but Arthur's Memory Cafe that I do here in Minnesota, we meet on the second and the fourth Wednesday of each month at 1 o'clock, and anyone around the world is invited to join us. Those are for people with dementia as well as their care partner. You can find more information on our Facebook page regarding that, or feel free just to reach out to me directly. And so that'll be this Wednesday, tomorrow the 14th. And then Artist Way in Minnesota is starting a memory cafe. We've done one already, but they are going to do theirs on the third Wednesday of each month from 1 to 2. And again, you can find more information on that, or you can call them directly to register at 612-200-0506. And let's see, we've got a couple of quizzes I want to tell you about. The first is a brain health quiz with MD V.I.P. It's a really interesting healthy brain survey, and I would highly recommend you take that. You can find both of these at alzheimerspeaks.com right on our homepage. The second was developed by a research student in New Zealand by the name of Chantel Horn, and her survey that she's doing is about dementia caregiving burnout, which, again, both of these are short. They're easy to do. And then, of course, I want to give a shout out to Coro Health, that's CORO Health, because they're still allowing people during COVID to download two of their apps. One is called Music First, and the other is called Choral Faith. And then, of course, the Memory Cafe. If you're looking for any other Memory Cafes, just go to memorycafedirectory.com. And if you go to Cafe Connect, that will bring you to all the virtual cafes. So let's hear from the Foot Bar Walker, and then we'll come back and talk with our guests.
Introducing the life-changing Foot Bar Walker.
I'm Peggy from Danville, Kentucky, and I'm 91 years old. The Foot Bar Walker revolutionized my care of George. The saving that I made from having to put him in a nursing home came to about $192,000. The Foot Bar Walker opens and closes just like a standard walker. The only thing that is different is the top bar and the foot bar. Does that ever make a difference?
Does someone you love use a walker? Do they struggle to get up from a seated position? Are you a caregiver dealing with physical pain and stress as you help your patient? The Foot Bar Walker was designed to assist not only the patient, but also the caregiver. Patients have more control standing up, and no lifting from the caregiver is required. See how it works at thefootbarwalker.com. That's thefootbarwalker.com. Peggy, would you recommend the Foot Bar Walker?
Do I ever? I would not be in the health that I'm in today at this age had it not been for the Foot Bar Walker.
And again, I can endorse that personally. I've seen it, touched it, and I have seen it in action, and it's absolutely fabulous. So I highly recommend it myself. Now, today we're going to be talking about research and giving you hope, and we're going to give you some updates on what INmune Bio and Imeka are both doing. So let me introduce you first to Dr. C.J. Barnum, who is the Vice President and Head of Neuroscience at INmune Bio, Inc. He is a neuroimmunologist with a PhD in neuroscience from Binghamton University. And he has a broad experience across neurodegenerative disease and psychiatric diseases, holding multiple positions in both academia and industry abroad. His focus has been on translating inflammation therapies into actual clinical treatments for neurological disease and using biomarkers to direct that approach. So welcome, CJ. How are you doing today?
Hi, Lori. Thank you for having me. I'm delighted to be here.
Well, I'm excited to have you with us. I'm just going to ask you really quick before I introduce your sidekick here. If you have been personally touched by dementia in your own family or circle of friends.
Yeah, so it's a great question, and in fact, I think all of us have to some degree. I've been relatively fortunate in the sense that I have not had a direct family member impacted recently, but I do have some very close friends who have family members impacted, and I can tell you the struggle is just, it's awful to watch, and we are just pedaling as fast as we can to try to get therapies to treat this disease.
Wonderful. Well, thank you. I'm going to put you on hold a second and introduce Dr. Maxime Descoteaux, and hopefully I'm not crucifying his name too bad. He is an international leader in medical imaging and diffusion MRI acquisition, reconstruction, visualization, and processing in many clinical applications. Max holds a PhD from INRIA, and he has a postdoctorate fellow at the Neurospin in Paris in computer science and neuroimaging, and he is also the CSO of Imeka. And so we're just thrilled to have him with us. He also serves as a full professor of computer science at the University of Sherbrooke in Canada, and has a research chair in the neuroinformatics area there. So welcome, Max. How are you doing today?
Great. Thanks for having me, Lori. It's a pleasure to be here.
And please go ahead and say your name properly if I really crucified it. You won't embarrass me.
No, my name is a Francophone name. Like I said, it means many wine hills, but we say Descoteaux, Maxime Descoteaux. But Max is fine. Thanks a lot.
Okay. Sounds good. Now, Max, I want to ask you as well if you've been personally touched by dementia in your circle of family and friends.
Yes, I have actually two great grandmother or uncles. I've suffered from Alzheimer's disease, so I've seen it around me and around my family and how devastating it can be. And a bit related, but my mom had multiple sclerosis, which is not Alzheimer's disease or dementia, but it is a neurodegenerative disease. So a lot of what we're going to say today can go beyond dementia and Alzheimer's disease. And so, yeah, it's been around my family and my surrounding.
Okay, wonderful. Well, thank you for sharing that. I'm going to go ahead and start out with CJ. And just so everybody knows, they've given me permission to call them by their first name, so we're just going to be a little informal here. So, CJ, can you tell us a little bit about the work being done at INmune Bio to fight Alzheimer's and dementia?
Yeah, thanks, Lori. So we have a little bit of a different approach than many of your listeners may be familiar with. We actually view the disease differently than the neurologist that you might see. So right now, the way the field views Alzheimer's as a neurological disease, we actually think the disease is an immunological disease that has symptoms of neurologic origin, right? So it starts with a dysfunctional immune system, and then it leads to these symptoms of Alzheimer's disease. And so our approach is to target the inflammatory dysfunction that we believe is underlying Alzheimer's disease in many patients.
Okay, wonderful. Now, you have a trial going on right now. Can you tell us what phase that is and who's involved, who's the target there, and what your findings are?
Yeah, great question. Thank you. So we have a phase 1 studies. So these are the very early studies where we're trying to do is demonstrate proof of biology. And what I mean by that is to make sure that our target is that we're actually hitting our target. And again, we're targeting inflammation. So what we're looking at is whether or not we can reduce inflammation in these patients. And what's interesting from our point of view, and I think what's different than what at least historically has been done in clinical trials in Alzheimer's, is we're using biomarkers to select patients. So our thought process is pretty simple. If you're targeting inflammation, your patients should have some evidence of inflammation. And so that's exactly what we do. So we are looking for patients that have Alzheimer's disease and biomarkers of inflammation. And one of the unique ways in which we have approached this is we've tried to make it clinically translatable. So we're not using very research-oriented or very tricky inflammatory biomarkers. We're using biomarkers that are done routinely at labs around the world. So specifically what we're looking at is if patients have a diagnosis of Alzheimer's, and they have an increase in a protein called C-reactive protein, which many of your patients may be familiar with. It's one of the panels that is done at the doctor's office all the time, which is a general broad marker of inflammation. Then patients are enrolled in that study. And at this stage, we're not looking to distinguish between mild, moderate, or severe patients, because again, what we're looking at is to show that we can see a reduction in neuroinflammation. Okay.
So is it just strictly people with the diagnosis of Alzheimer's? No, nothing Lewy body, vascular, any other types of dementia?
Correct. So at this stage, we are selecting patients based on a clinical diagnosis and not a biological diagnosis. So the extent to which patients may have, Lewy bodies or any of these other pathological symptoms is not something we're exploring in detail at this point. So it's just a clinical diagnosis. So I expect there'll be some patients in the trial that may not have full Alzheimer's and may have some other pathologies as well.
Okay, yeah, because I know a lot of times things switch, as the disease progresses, and and as technology and our doctors out there get more sophisticated and knowledgeable with things as well. So whereabouts are you at with this clinical study? Is it closed and you're just, wrapping up your research, or is it still an open study, this phase 1 that you're doing?
Yeah, so we are in the process of finalizing the study. As you might imagine, coronavirus has stalled it a little bit, but we expect to be completed with the study by the end of the year. And from then, we anticipate opening a phase 2 study sometime mid-next year, and hopefully this will be in the United States. Right now, our study is being done in Australia, and and we expect to bring that to the United States for phase 2. Okay.
Sounds great. I'm just going to pull Max in and see if he's got any comments regarding what you've stated or anything that you want to toss in there, Max.
No, this is pretty complete. I'm more on the technology side, on how we're about to image this inflammation. So, I guess we'll be discussing this later.
Okay. Well, and later is coming right now. So what I want you to first tell us a little bit about Imeka. Who are you? What are you doing? And why are you there?
Yeah. So Imeka it used to be a startup. Now it's a company of 13 employees based in Canada, Sherbrooke. You mentioned Sherbrooke, which is about an hour away from Vermont and 4 hours away from Boston. Where there's a great university, and this is where I started my lab. And part of the technology from my lab has gone and spun off into Imeka. And so I did my whole PhD and most of my research career working on brain imaging and finding technology, and especially a non-invasive technology just based on MRI. So MRI is magnetic resonance imaging, that usual kind of tunnel machine that people have seen on the news, and use this MRI machine to try to non-invasively image brain tissue and brain microstructure to try to provide what I like to call like a virtual dissection of the brain without having to open up the brain and take tissue and biopsies and put that under the microscope. We're developing a technology that's able to image tissue and quantify what's going on, so we can then work with companies like INmune Bio and see what is the effect of a drug, or what is the effect of a disease, or anything on brain tissue, and in particular white matter. So Imeka is really specialized into matter imaging at the service of pharmaceutical trial and biotechnology to provide a cutting-edge solution to quantify white matter tissue.
Okay. Well, I love that it's non-invasive. I mean, everyone's thrilled to hear that, to not have to go under the knife and be able to get that like you said, virtual imaging at all different angles to really see what's going on. One question I have for you, though, have you run into, because I've heard this from not just people with dementia, but a lot of people with dementia, where they're just not comfortable going into the tube, to have that imaging done. In order to still get valid pictures of all this, can somebody be sedated to go in to the machine? And
I believe this is unethical. I don't think we do this.
Okay.
We may do so under very special circumstances, maybe for certain Alzheimer's patients, because they're scared they're going to be moving or have an anxiety attack in that tube. It's true that it is a pretty tight tube and can be a bit claustrophobic when you're in there, but usually we don't have that many issues with the actual imaging inside the tube. People kind of get used to it and trust that what we are imaging is actually worth a lot, so they concentrate, and we try to maintain our sequences or the exam quite short, to spend as least amount of time as possible inside the tube. So it's usually, in between 10 to 20 minutes.
Okay.
For clinical trials.
Okay. The reason I asked it is, my mom had some MRIs done, and it had nothing to do with her dementia. She was in a car accident and had a brain bleed, and they needed to sedate her in order to keep her still because she's very claustrophobic. Right. And I've just heard that from other people, or the pinging noise. I don't know if that still exists. That happens during the MRI.
It's noisy. It is noisy. Yeah.
Yeah, and I've heard some people have used like earplugs so that they don't hear that pinging. Other people with dementia have asked about could they use like a headset, and I don't know what could be used or what might interfere with the whole technology. That's why I'm asking these questions.
Well, if I may just complete my answer, but this is done by default. So head plugs, earphones, and also a lot of cushions are placed around the head just to try to attenuate these sounds. So it's much less noisy. Usually we can also put music. We can put a little, soft music to try to calm people down. So I know that even when we do controls or participants that don't have any disease, we usually use these tricks as well.
Mm-hmm. Yeah. Oh, great idea.
This helps.
I'm glad that is okay and doesn't interfere. And I know that so many people, they want to push research forward, and they will fight tooth and nail to be able to be part of these studies. Even if it's uncomfortable for them, because they think this is really important information for us to gain for the future of the disease and stuff. Now, a lot of people might not know what white matter is, so I'm going to have you just explain that, if you wouldn't mind, for us, Max.
So I'll try to give you a kind of an image or a mental picture. So the cell that's critical for the nervous system is the neuron, and the neuron lives in gray matter. So I think people have heard about gray matter, and gray matter is mostly on the outskirts of your brain. And so I like to compare the neuron to, let's say, our cities. So the big cities would be the little neurons everywhere spread out in our brain, and what connects these neurons together is the white matter. It's the axons, if you wish, the roads in between the cities, or the wiring diagram of our brain that connect different neurons together and that assure transmission of information, electrical information, through the brain. And so this is incredibly complicated. We estimate that we have more or less 85 billion neurons. This is huge. And about 100,000 miles of white matter. That is, axons and wires connecting different neurons together. And so this is the white matter. It's a huge part of our brain, mostly within our brain, and just, wiring different neurons together to make sure that, vision is talking to motor, and motor is talking to memory, and memory is talking to language, and on like that. So it's imaging white matter and quantifying white matter is really, truly trying to map out the wiring diagram of the brain, or if you wish, the Google map of our brain. So It's crucial to understanding anything about the brain.
Okay. Now, I have this question before me, and I don't have a clue what it means, so I'm just going to throw it out there and have you define this for us. But what is free water imaging, and how does your company apply that technology? Does it differ from the MRI, or is that just part of the technology? I'm not sure on that one.
Yeah, it's part of the technology. If you wish, imagine again analogy with the roads, and if you imagine you're on a road which has a huge pothole or construction site, so a big roadblock, and that could be from flooding. For instance, comparing this to free water, it could be from excessive rain, and suddenly there's flooding, and some roads are flooded with extra water on them. Free water imaging is actually trying to quantify how much of this water is actually contaminating the road. And the analogy is quite powerful. It's something similar. That's what we're doing with imaging. So when we're trying to reconstruct these 100,000 miles of axons and white matter, anywhere there is abnormal white matter or abnormal wiring with more liquid, more water around the tissue, well, our technology is sensitive to pick it up. So we can actually turn this into a number or into a map where you see hotspots or alterations of this number. A bit like if you wish you had satellites taking images of our roads, and you could highlight where are the roads blocked and where are the roads flooded. And so using this technology and non-invasive diffusion MRI, and the word diffusion says it, so it's this water that we're trying to quantify with diffusion MRI. Then we can turn this into healthy-looking roads or diseased-looking roads with some inflammation on them. So inflammation means more extracellular water, and so more extracellular water is this free water term that we like to use now. And so this is our, if you wish, indirect marker of neuroinflammation.
Mm-hmm. Okay. Well, that helps explain it. I like your analogies. It's easy to figure out a pothole and how that affects our lives and stuff.
Yeah,
so,
and how it affects traffic along this road. And the traffic here is your electrical signals navigating in these wires, trying to connect different neurons together.
Exactly. Now, what can you tell us about the latest results from INmune Bio's clinical trial on Alzheimer's patients?
Well, what we can tell you is that, well, of course, this is preliminary. We don't have all the data acquired yet. So this is on a subset of participants. But what we see is clearly very strong trends that something is happening. Huge changes in this free water marker. So reduction in free water from things I've never seen in experimental data in the past. So each participant, it's his own control. That is, we have a baseline image before treatment, and we have different time points once the treatment has started with different doses. And what we see is under different fiber pathways, these white matter pathways, there is a clear reduction in free water overall, but also in particular on the language system. And so that attracted a lot of our interest. And at first sight, it wasn't an obvious candidate. Why language and not, for instance, memory or another system? But what we seem to see in these preliminary results is a clear reduction of inflammation on the language system, which we call the arcuate fasciculus. So this is very encouraging, and we can't wait to have more of the participants' data to really have robust statistical tests to confirm these preliminary findings.
Wow, that is exciting. So in kind of talking language that my listeners may or may not understand, aphasia really affects people's speech. Would this be one of those symptoms that could maybe be controlled a little bit more, or the symptoms reduced due to this? Exactly.
And if CJ wants to jump in here, we do have also a neurologist in Australia that explained to us what's happening in these patients, and we see also clear clinical manifestation of improvement of some of their language skills or everyday daily activities, and CJ is in better position than me to comment on this, but this is clearly very encouraging.
Okay, CJ, do you want to comment on that?
Yeah, thanks, Max. It's interesting. The way that we have measured inflammation in the past is we would have to do a lumbar puncture and collect cerebral spinal fluid, which has been very invasive, and it tells us a little bit about inflammation, but it doesn't tell us where. the real, and Max is being modest here, the real power in their technology is that not only can you measure it in a way that's non-invasive, but you can see exactly where it's happening. And to his comment earlier, what we found is in the very first 6 patients that we analyzed, a very large reduction in inflammation in the brain, and we could pinpoint exactly where that was occurring. And because it was occurring in a place that's important for Alzheimer's disease, and we could see changes there, and by the way, this is over a 12-week period, so a very short period of time, is just phenomenal as it relates to developing new technologies to assess therapies in Alzheimer's disease. And so this is one of the things that we're trying to explore further. To what Max was saying, we had a webinar where we discussed our preclinical findings, or this interim data from the 6 patients, and that whole webinar can be viewed at our website. But what he was referring to was the clinician came on ahead of time that has been treating most of these patients, and she just said some really remarkable changes in the patients that have more mild disease, and this really did correlate with the changes that we saw in the free water. So we think this is going to be a really powerful tool, and it really does allow us to look at things in a way that have never been looked at before. So one of the things, and I'll let Max elaborate on this, is not only can they measure neuroinflammation, which is what we're interested in, but they can also measure whether or not these axons, this white matter, is changing over time. And there's a few key elements in there. But these white matter changes actually occur pretty early in the course of the disease. And once they occur, that's when people start having cognitive problems. And so we think that this is going to be a much better measure because we're measuring the roads built as opposed to the cells that are lost. Once the cells have died, we don't think we need a different type of therapy. These are regenerative therapies that you need. But as long as we can track the white matter changes and show that therapies are improving white matter, rebuilding those roads, then the connections between different parts of the brain can continue and reestablish. And we think this will be a much more sensitive measure. And Max's company has the ability to not only look at the inflammation, the potholes per se, but to see whether or not you're building new roads and how those roads are being built. And we think this is just a really powerful technology and an advancement in how we're going to assess patients in these trials. Because we can look at this in a very short period of time.
Mm-hmm. Well, that is fascinating. When you said 12 weeks, I just about fell off my chair because that really is astounding that you can see a difference in that short of a time frame. And then for it to be, non-invasive. I want to put Max in here for a second because, again, it sounds like he was being pretty modest in terms of what he's developed here. And I'm wondering, in terms of your MRI testing, is that something that is available now to people, or is it only through the clinical trials that people can take advantage of your imaging?
Thanks for the question. That's a tough one. It's let's say the way the images are acquired is available on almost any machine in research hospital centers across the world. And this is a reason why, for instance, we can work with Australia and almost with any hospital in the world. What's complicated is the image processing that goes on behind, which is still unavailable easily. That's why a big reason why the company exists and my lab still exists, where we do on our daily life is to improve and develop these software and processing techniques to quantify these images. And so it's a bit of a yes and no to your question.
Okay.
What's most commonly available, I would say, is for surgeries. So neurosurgeons do have access to this brain mapping technology to see the white matter around a brain tumor, for instance. And that's available to them, and that's been FDA approved, and they can use it in the operation room. But I would say for almost everything else in the clinic, it's not routinely available yet. We're actively working on this, try to, close the gap and make it more translated to clinical routine.
Okay, well that makes sense. I mean, I just noticed, and I'm by far no brain surgeon or close to where you guys are in terms of things, but just in terms of general technology, you can have it, but that doesn't mean you know how to use it. And I think everyone's felt that through different things that they've downloaded over time, or, I can do this much of it.
It's with trials like the one we're describing now with INmune that hopefully in the future we can convince, the FDA and Health Canada that this should be incorporated as almost as a default when you do brain imaging. And I hope personally that's a clear goal in my career. If I can do this within the next, 10, 15, 20 years, that would be a big mission accomplished in terms of translating the technology to everybody.
Oh, that would be wonderful, and it would give people so much hope and insight at an earlier stage. And again, not being being non-invasive is a huge factor. I'm going to ask you a silly question, but I always think of inflammation attached to sugar. And do you see anything within your studies in terms of maybe having people go off of sugar during this as well, or am I just left field on this? And I'm going to throw this to CJ first and then come back to Mac.
So, Lori, you are spot on. In fact, what I find interesting, and, this is just between you and me, what I find is the non-scientists understand inflammation better than the scientists that study anything but inflammation. That sort of intuitively understand it. And yes, in fact, the link between sugar and diet and exercise is really, really strong. In fact, one of the biomarkers that we use in our clinical trials that will allow people to get in, besides that C-reactive protein that I was telling you about, is hemoglobin A1C. And for those of you that don't know what hemoglobin A1C is, it's typically a marker of diabetes. But we know that people that have elevated hemoglobin A1C or have diabetes have a very strong inflammatory drive. And we also know that anti-inflammatories do a really good job of reducing symptoms in patients that have elevated hemoglobin A1C. So to your point, diet is really critical. Diet and exercise are very, very critical to overall health, brain health. And a lot of that is because of its effects on inflammation. So the extent to which you can have a very good, healthy diet, that you're exercising, those things will go a long way throughout life to keep your inflammation at bay.
Okay, great. Max, anything that you wanted to add in to my question about sugar at all?
I could, a little maybe opening a parenthesis, but we had and CJ was there last week, a webinar organized by Imeka where we can actually image, again with brain imaging, how the brain consumes glucose, which is sugar, and how it consumes other things. And one of the hypotheses around is that maybe our neurons are not dead, or the cells are not dead yet, but they just have a hard time consuming that usual sugar that is the primary fuel. And when you start to try therapies such as, ketogenic diet that try to bring in complementary fuel to the brain, well, we actually can see that the white matter and the gray matter uses that fuel differently and may help certain white matter and gray matter tissue to do better. And so this is really cutting edge, exciting research going on at the moment. That's a bit of related to the question you just asked.
Okay, great. Thank you. CJ, with your second trial coming up here in 2021, do you see it expanding at all to include diet and exercise as well, or are you still in the initial stages of we've got to get this part done first with the imaging and get a better handle on that?
So that's a great question. We don't put any restrictions in as it relates to not being able to do it. When you start adding different elements into it, it makes the trial really big and difficult to enroll patients, because you have to. The idea of the clinical program is to control for variables to make sure that the effect of your drug is really the effect of your drug and not something else. So if we include that, then it gets really big and it's hard to enroll, and then you got to sort of tease it apart. So that's not something that will start right away. But that doesn't mean that we don't keep patients from starting an exercise program and eating right. In fact, we encourage those things. I think for the patients that we're talking about, once you start getting to the stage where you're more mild or moderate, those things will help. But at that point, the inflammation that you have, and I like to call it immune dysfunction, a dysfunctional immune system, probably needs a stronger solution. And so we think that in combination, these things will work well, but you're definitely going to need something that targets the inflammation more directly.
Okay, that makes sense, and I can see how that would complicate things, but I just thought, I'll ask. And one of the questions that I know I hear, comments I should say, that I hear from people enrolled in these studies is, I got kicked out because of this, and I got kicked out because of that. And do people, let's say they're diagnosed with Alzheimer's disease, but maybe they have something else, maybe they have diabetes or a heart condition, is that going to kick them out of the study? What kind of criteria do you have, CJ?
Yeah, so each clinical trial, the criteria is going to be a little bit different. And part of it is to try to keep the number of variables low so that we can really, so when we get to the end of the study, we know that it's our drug and not something else, right? So a good example of this is if you're in the middle of your clinical trial, you start taking two other drugs for whatever reason, and then you have, an adverse event. We call it an adverse event. Maybe you have some, it could be something as serious as a heart attack, or it could be a reaction. Then we don't know whether or not that's the drug, our drug, or whether or not it's another drug. So what we try to say is during the clinical trials that patients need to be stable in their medications, right? So we know that nothing's changed over time. And usually there's some time frame before and after in which that's the case. Mostly 60 to 90 days they need to be stable, which means they need to have been on the drug that they're coming in on for 90 days. The other thing that is taken into account is how your drug may interact with drugs out there, right? So there are many drugs that get broken down in the body, this is an example, by the same mechanisms. And when those drugs are put together, what happens is your body has to sort of compete for that, and you get side effects. So some of these medicines are looking for drug interactions, and you may not be able to be on a drug because of those interactions, right? So that may be another thing. From our perspective, for us, our drug is pretty unique, at least in the way that it works. It's highly specific, and we have not identified any drug interactions. And the other part this gets to is some of those other elements you're talking about, if they have diabetes or they have this. And a lot of times clinical trials will exclude those patients because they have other comorbid disease. Actually, for us, those are the patients that we want. It turns out that when you have inflammation as sort of this underlying pathology, you tend to have more comorbidities. In fact, when I'm talking to somebody, let's say I'm talking to a physician, they say, all right, well, tell me what a patient with inflammation looks like. And I have a couple different criteria. My first one is do they have symptoms that don't really fit into a specific bucket, right? So they've got a disease, but they've also got these other things. And if they say yes, then I say, do they have multiple comorbidities? Oh, yes. And then the third thing is what do their blood inflammation levels look like? And typically those things all point to someone that has some sort of underlying inflammation. And the reason for that is when your immune system becomes dysfunctional, it goes after your weakest biological link. And that's different for everybody. So if you have 100 people that have immune dysfunction, one person's biological link may be a predisposition towards depression. Another person may be diabetes. And so that's sort of this underlying pathology of inflammation. And so for us, we are happy to take the patients that have these comorbidities. In fact, there are very few comorbidities that we don't allow into the study. Some of them are you sort of have to have. Like, someone has a life-threatening disease like active cancer. That's a different story, or an active infection. But for the most part, we have very flexible criteria as it relates to bringing people in the study.
Okay, wonderful. Thank you. I'm just going to ask Max if there's anything you'd like to add.
No, this is good for me.
Okay. Well, I really appreciate the time you guys have spent with us today. And again, I want to thank Dr. C.J. Barnum and Dr. I'm going to kill your name, Max, one more time. Dr. Descoteaux. I'm going to have you say it because I'm so paranoid I'm going to screw it up. So, Max, say your last name again for me.
Descoteaux.
Okay, thank you. For our listeners, you can reach out to either of these individuals. For CJ, you can go to their website at INmuneBio.com. INmuneBio.com. And if you'd like to email, we're going to direct emails to D. Moss, that's David Moss, the CFO, at INmuneBio.com. And they are also on Twitter. You can find them there as well as LinkedIn. And then for Max, you can shoot him an email with any questions you have at M. And then I'm going to spell his name so you'll know why I was tripping over it. DESCOTEAUX at Imeka.ca. And again, their website is Imeka.ca. They're also on Twitter and also on LinkedIn. So I want to again thank everybody so much for the show today. I found it very interesting, as I'm sure our listeners did. You can always go to Alzheimer Speaks to find out more about our work. Have a blessed week, everyone. Bye now.
It's time to rethink, renew, and reimagine retirement. Hey everybody, Jared Sylvester here, host of Retire Repurposed. Now this podcast is about the non-financial parts of retirement, which many times can be even more challenging than the financial. We believe retirement is not the end, rather the beginning of what could be the most impactful, purposeful, and fulfilling season of a person's life. So don't retire, become repurposed. To listen now, search Retire Repurposed on your favorite podcast platform, Senior Resource, or Life Audio.